Welcome to this Cell and Gene Therapy Insights Fast Facts with C Technologies, a Repligen company. We'll be hearing from Ramzi Shambaki, Bioprocess Applications Manager, as he discusses a novel approach to at-line and in-line concentration measurements for process development and GMP manufacture of biotherapeutics. Ramzi spent the first 8 years of his career working in R&D, manufacturing and product management, designing and building fiber optic products for high-powered laser delivery in the medical device industry. For the past 5 years, he's been partnering with customers to implement the FlowVPE system for inline concentration measurements in both process development and GMP manufacturing settings. Over to you, Ramzi.
Hi, this is Ramzi Shambaki. I'm the Applications Manager for C Technologies, a Repligen company. We're the analytics division of Repligen, and we have the SoloVPE and FlowVPE products. Today, I'm here to talk to you about how these products can save you a lot of time in the gene therapy workflow, measuring concentration, both in process development and in manufacturing. Okay, let's get started. Today, we're going to start with some explanation about traditional UV spectroscopy and how we've had challenges over the many years of use. We'll detail how slope spectroscopy method works, some advantages there, and the at-line and in-line solutions that we provide, and then some real-world examples in the gene therapy market of using these products on real gene therapy samples. And then I'll explain how C-Tech goes about their business and how we can support you much better throughout your process development. C-Tech is part of the Repligen Corporation. We represent the SoloVPE and FlowVPE systems for measuring concentration. Our claim to fame is with the protein concentration or MAB concentration. But more recently, we've been working in the gene therapy market to look at AAV, lenti, and other cell therapies. Repligen has been around since 1985, and they have various suite of products, Opus PD columns, ATF-XL for harvest intensification, and then TFF systems and flat sheet cassettes, along with hollow fiber filtration systems. So traditional UV spectroscopy, it goes back maybe 50 years. It's based off of a standard spectrophotometer that would read wavelengths from the range of, say, 200 to 900 nanometers. It's based off of standard cuvette that's 1 centimeter. And what the issue is when you have samples that are out of the range of the spectrophotometer, you have to then dilute the sample or somehow treat the sample so that the UV spectrophotometer can get a solid reading. In this example, you can see the light goes through the side of the cuvette. The path length is defined by the distance between the walls of the cuvette, and it's a fixed value at 1 centimeter. The analysis to get concentration is based off of Beer's law. Absorbance equals extinction coefficient times path length times concentration. And in this case, the concentration is the variable, which is what you're actually trying to look for. So there's challenges with standard spectrophotometry. The first being that when you have to do sample prep, you have to do careful manipulation of the sample that sometimes requires a long time. It can be anywhere from 30 minutes to 3 hours. During this time, you might have a hold step in production where you are not able to move forward the process because you're waiting for samples from your QC or analytical lab. The manual manipulation of the sample also increases the opportunity for error to occur in your measurement. And then we've seen dilution error in some cases as bad as 20%, but even in simple cases as low as 5%, which is still not really acceptable. In contrast to traditional UV spectroscopy, the SoloVPE and FlowVPE products use slope spectroscopy. This is where the sample will stay the same, and instead we vary the path length with a precision linear stage. In this case, we now only have to worry about measuring and reporting the data rather than dilution, measure, plot, calculate, and many other steps that are required for the standard traditional UV spectroscopy. So we, in effect, automate the time of dilution and give you the answer right away in a robust and simple manner. Like traditional UV spectroscopy, slope spectroscopy is also based off of Beer's law. In this case, we use the path length as the variable that's controlled by the system, and your concentration can remain constant, thus not requiring the need for a dilution. We also use multiple path lengths during the measurement. In this example, 10 path lengths that will then quantify your concentration very accurately and in a robust manner, rather than a single data point that could be lost due to some error or a smudge on the cuvette. So how does slope spectroscopy work? Well, the system has a fiber optic piece called a fibrette that carries the light from a spectrophotometer to the sample. That is connected to a precision linear stage that can vary path lengths anywhere from 5 microns all the way to 15 millimeters. It has an automated algorithm we call quick slope that lowers the fibrette to the 0 path length, finds 1 absorbance, and then will collect 10 data points based off of numbers below 1 absorbance. So you can always be sure that you have a linear regression to your concentration. We also output the R-squared value, which is given as 0.999 or higher to be sure that you have highly linear and accurate data. In the bottom right-hand corner, you can see an example of SoloVPE quick slope results, where we've quantified the slope as 28, which equates to a concentration of 203 mg per mil. Some advantages of the VPE system are displayed here. One of the main advantages is the wide linear range, and that's due to the path length range that's offered with the system. This is true for both the SoloVPE, the at-line system, and the FlowVPE, the inline system. You can go from a range of about 0.01 to 300 mg per mil. Because of the multiple data point reads, we have very good repeatability of plus or minus 2%. We also can take a measurement in about 1 minute. So in a short amount of time, you can get your concentration rather than taking a long time in offline measurements and complicated analysis. On the inline system, you can get measurements every 5 seconds, meaning that you could use it even for the fastest chromatography processes. On the Solo, the sample requirements are rather low. If you have enough concentration, it takes about 20 microliters. And then on the FlowVPE, you have multiple different flow cells to fit any size process. In the approximate 10-year history of the VPE products, we have over 1,300 systems installed and validated this method on many, many different drug products or substances across all of biopharma. We have multiple publications that you can get off of our website and a lot of information on how to actually implement this technology. There are many different applications for the SoloVPE technology, starting with our number 1, of course, protein concentration at A280. This is an application that's widely used across biopharma, both in the early stage development all the way through product release. We can also do polysorbate analysis, integrity testing for your filters, and DNA and RNA analysis, which applies directly to the gene and cell therapy market. And other applications like antibody-drug conjugates or multi-component analysis for blood products. All the benefits are, like I said before, no dilutions, no sample manipulation, the wide dynamic range, the robust measurements, which ultimately results in reducing the production hold time and directly getting a measurement as you're making your product in the production floor. So I just talked about the SoloVPE system for at-line analysis, and I'd like to introduce the FlowVPE system, which is for in-line analysis. On this system, it uses the same slope spectroscopy technique, except you can use it in-line during the downstream process. It's particularly strong in UFDF and drug product, but can also be used to measure high molecular weight species in your polishing step or for load and elution in your capture step. Again, no more sampling means no more long wait times. It also means saving material that you would normally use for sampling, which is very precious in the gene therapy market. And it gives you actionable data that you can then connect to your process and control the process, bringing you better efficiency to your process. So in this example, I'm going to talk a little bit about how the slope spectroscopy technique can be used to quantify your DNA, RNA concentration and purity. In this drawing, you can see that there are different peaks that are associated with the different types of materials in your product, with the peak at 260 representing the DNA and RNA, and the shoulder at 280 representing the protein. One study we worked on was with Pfizer, where we looked at 25 different mixes of plasma and insulin and measured the ratio of A260 to 280 to get the R value or purity value of the sample. We've released an application note that describes this work, and you can see some of it on the screen here, where we've measured multiple different samples and compared them to the offline measurements with very good accuracy. Using the FlowVPE system, you can put it in line with your UFDF process as you're concentrating up your AAV. It can be used, again, measuring A260 and 280 together to give you the concentration of your full capsids as it's rising through the process. You can also look at the diafiltration and make sure that you have a steady signal there. In this example, we've compared to the offline ddPCR with very good comparison. We also had a situation where we noticed the sample getting cloudy as it was being mixed, and that corresponded to a rise in signal from the FlowVPE. So this is a possibility that you might be able to make an action on your process to potentially improve the quality by carefully looking at the signal from the FlowVPE. As I mentioned before, our products are very strong in the antibody processing market. Various customers have used the SoloVPE and FlowVPE for many years and have been able to validate that method in their process. In all cases, they've saved multiple days of man hours using the SoloVPE system in the production suite. And they've realized great repeatability on their data, regardless of the concentration range. It could be very low concentration or even very high concentration. So with VPE technology, we're taking away the 7 or 8 steps that it might take for you to do a traditional UV spectroscopy, down to 1 step for the SoloVPE, or no steps with the FlowVPE. And the result is built-in data quality, easy to validate for GMP, simply measure and report and get your data right on time. We're committed to our customers' success, not only with the products that we offer, but also the services that we offer along with the systems. We offer validation services and supports, which could include a computer system validation, IQ, OQ of your system. We also will help during the implementation process to guide you through 21 CFR Part 11 compliance, and then validation support on your method validation. In addition, we can also help integrating your systems. If you have a FlowVPE system and you want to integrate it to another skid, we can help with that. We can also look at your SOP documents and provide review and improvement upon those from the vast experience that we've had serving our customers in the biopharma market. So thanks for listening. If you want to learn more, you can hop onto our website at ctechnologiesinc.com. We have vast amounts of literature about the products that you can read for yourself. If you want to get more detailed information specific to your process, please contact us at the /contact site, and we can have one of our local reps contact you.
Thank you, Ramsey, for a great presentation. And don't forget, you can post your questions to Ramsey using the Ask a Question form on this page. Thank you very much for joining us today.